Yeah.
Okay.
So the bell rang and it's 10 o'clock exactly. Good day, everybody, and thank you for coming again to our ZOOMinar series. I want to make a short announcement that the abstracts for the ECR presentations have been selected. There will be 9 presentations, and we split them into 2. They will be on Wednesdays from 9 a.m. to 12 p.m. on June 23 and July 14th. So please mark your calendars. It will be great to see you all there to hear from the young researchers. And before we start with the seminar series, I want to remind everybody that we will have 2 speakers who will speak about for 30 minutes. After the first speaker will be about 10 minutes questions. And after the second will be again directed to the first speaker and another 10-minute questions at the end of the second speaker. And after that, we will have general discussions where we will allow everybody into the panel to ask their questions personally. And I don't want to take more time, and I'm passing you to you, Claudio.
Yes. So thanks for asking me to do this. I'm very happy to introduce Lucia. I think she is known to most of you, but nevertheless, Lucia is presently director of CHERM. She's been director in the past for, I think, 6 years, and then we took a shift. So I was director for a little while, and then Lucia is back. And you know CHERM. CHERM is a Center for Magnetic Resonance in Florence, and Lucia is one of the founders. Lucia graduated in '78 and in chemistry, and she spent several periods abroad, but then she made all her career at the University of Florence, where she became full professor in 1999. And she is now, besides being director of CHERM, she is the head of the Italian core center of the ESTRI research infrastructure Erc, which is one of the European infrastructure. Erc stands for European Research Infrastructure Consortium, which is one of the infrastructures that is localized over several centers, and CHERM, and she as the head of this is the main NMR center in this structural biology infrastructure. She is also a member of the Erc Executive Committee and of the Council. So I may say that she's actually, if not the soul, but one of the souls of this infrastructure, which is being very successful and is helping a lot of people to actually address problems of structural biology from multiple points of view. Lucia has been working mostly using NMR, and she's well known for structural biology. She has applied NMR, developed NMR techniques for structural biology over many years. And more recently, she's addressing structural biology in cells. So she's actually doing NMR in cells and discovering a lot of interesting things that one could not see or could not even expect in solution. So it's a kind of a breakthrough, and I think she will tell you about it in her lecture. And she's, of course, both of us are originally inorganic chemists, so we work on metalloproteins, and she's been particularly addressing the issue of how metals like copper and iron are transported, delivered, and used in cells. So in-cell NMR is actually one way, a very good way to actually look at this phenomenon. And she's published about 400 articles with more than 20,000 citations. And she has received a number of prizes, which I will read. One is the IUPAC Distinguished Women in Chemistry in 2015, and the gold medal of the city of Florence, which is a local recognition, but it's a very important one, also in 2015. Then she received the Instruct is the infrastructure, Instruct Award, called Bertini Award, in the name of Ivano Bertini, in 2018, and she's a member of EMBO, European Organization for Molecular Biology, and the Italian delegate at the Council of the European Molecular Biology Laboratory, EMBL. I think I will leave the floor to Lucia without taking much more time. Please, Lucia.
Okay. Thanks, Claudio, and thanks, Rams and all the organizers to give me the possibility to present this and to have organized this series that is very interesting. I participated in a few of them, listened to a few of them are very interesting. And within the frame of this, the spirit of this series, I selected a few examples of the application of in-cell NMR, as Claudio mentioned, is something we have recently developed and exploited a specific approach in in-cell NMR in CHERM. And the specific example will be focused on protein folding and maturation, which is quite relevant with respect to neurological diseases where you have protein aggregation and misfolding. Just a clarification, I think the audience knows what is in-cell NMR is, but just to mention that what we mean is detecting, applying a high resolution NMR for the detection of proteins or biomolecules inside living cells. So what we put in the NMR tube are living cells which have proteins that are labeled, selectively labeled or uniformly labeled. And there are several approaches and types for preparing these samples and to have labeled materials inside living cells. And I mean, the idea of in-cell NMR has been initially developed, proposed by Volker Deutsch, and then exploited by a number of different, several scientists from Shirakawa, Pilak, Selenko. I don't want to, I forgot some of them for sure, but what has been used by these various people is either protein expression directly in a bacterial cell, like E. coli, that is simple, or if you want to use eukaryotic cells and mammalian cells, just they were inserted the protein by microinjection or exploiting different techniques to internalize the protein. What we have developed in Florence is an efficient way, a good way to have good level of and almost selective labeling of the protein or proteins of interest inside mammalian cell. These are stabilized human cells, directly in the cells which we put in the NMR tube. And this allows the characterization of proteins since even the early steps of their productions and or the folding and the effect of different materials or treatment with respect to, for example, the maturation folding. And one example I would like to show the application of this approach and how we think is powerful is related to copper transport, and with particular focus on the incorporation of copper in SOD. And the reason why, I mean, we have characterized this because we have worked on SOD since ever, but also, you know that SOD has involvement in neurological diseases, so that it fits in this type of series. And superoxide dismutase is an enzyme which has the role of produce dismutation of superoxide radicals in oxygen and nitrogen peroxide. And this enzyme, once is synthesized by the ribosome, has to undergo a series of post-translational modification, going from the zinc binding and then the copper binding which is the site for the enzymatic reaction, disulfide bond formation, which is essential for protein stability and somehow prevention of aggregation, and dimerization. So the question we have is that how these steps occur in the cell, because in vitro you can produce and you can produce any of these intermediate with any variable order. So that is very simple to do this. But then in the cell, what is happening? And for this, as I said, we develop this approach where we effectively produce a reasonable level of the protein inside the cell, human cells, and then we put these cells in the NMR tube and they will acquire the NMR spectra. And so in this specific case of SOD, if cells are grown without extradiction of zinc, what we obtain is an essentially unstructured protein, has the apo form, the metal-free should be, and if we use, just to increase the resolution, we selectively label the cysteines, we can see that the cysteines are all in the reduced state, so there is no oxidation of the cysteines. If we grow cells in presence of zinc, we obtain a fully folded protein, and I would like that you appreciate the quality, the resolution of this spectrum, which seems like a solution HMQC spectrum. And so it's fully folded, and from the analysis of the spectrum and the comparison with the various states of the protein in vitro, which were completely assigned, we can tell that this protein is dimeric, has a zinc bound only into the zinc binding site. And again, with selective labeling on cysteine, we can see that the cysteines are all in the reduced state. When we expose the cell to copper, copper is only partially bound and uptaken to the cell. And this makes sense because copper is very toxic, and we have systems to control the and block the uptake, let's say, to have control of the uptake of copper. And indeed, it's known that for copper incorporation to this enzyme, there is a specific chaperone which transport copper and insert copper selectively to this protein. And indeed, as we have slightly overexpressed the protein in this cell, the partner protein is not present in similar level. This copper chaperone is called copper chaperone for SOD CCS, which has been discovered by Valcolot and Tom O'Halloran and was called the 2A. What we found is also responsible of the formation of a disulfide bond. And indeed, there is, this is still in the reduced state. If we co-express similar level of this protein, this chaperone CCS, we have a simultaneous incorporation of copper, which in the cell is only copper 1, and we have the full formation of disulfide bond. So we have still a folded protein. We recognize that this is with a copper bound, and the cysteine are all in the reduced state. So then we can conclude that the steps are this way. They follow this path. Zinc needs to be incorporated first, and then is followed by the insertion of copper, has copper 1, together with the formation of disulfide bonds, which stabilize the structure. And this is both of these two steps are promoted by CCS. So having these tools for characterizing this in details, these processes in the cell, we analyze a series of SOD mutants. As I mentioned, I mean, and for sure you know that mutations of SOD has been related to the familiar form of ALS, amyotrophic lateral sclerosis. And these mutations are distributed, spread over all the structure of the entire sequence of the protein. And what has been proposed is that these mutations do not have effect on, do not affect the function of the enzyme, but affect the stability of the enzyme, and because they partially impair the maturation of SOD and the stability of the protein itself, which then tends to aggregate. Even the wild type, when is lacking the zinc and when is lacking the disulfide bond, tends to aggregate by itself as well. The wild type SOD aggregates like the other mutants with different rates, but they all aggregate. So we produce some of these mutants and we characterize them in cell. And you can see that here there is a kind of misfolded, kind of molten globule state and form. And these mutants do not bind zinc in the cell, even if we have added a lot of amount of zinc to the culture. And so they do not evolve towards a native form. And indeed, this type of molten globule state might be the precursor of SOD aggregates. What is peculiar is that these some of these mutations do not affect zinc binding in vitro. And if we treat these proteins with zinc in vitro, we obtain a fully folded form. So this also points out to the fact that this behavior is totally different if we analyze just in the buffer or if we analyze in the cell, in the cytoplasmal cell, where there are all the various components. So this is a very important message that really we need to characterize functional states of protein in conditions as close as possible to the physiological ones. And so then what we found is that if we overexpress CCS at similar level, then CCS contribute also to the uptake of zinc. So in this respect, CCS acts as a molecular chaperone. And so then when CCS is a comparable level, then this promotes the correct pathway to correct maturation of the protein, and then prevents the formation of these molten globule states, which then tends to aggregate. So this also gives information that the altered pathway in for the SOD and the aggregation might be is connected to the altered maturation of SOD. Just to go through some other examples which are relevant for protein stability and the specific functional states, we have characterized, for example, the oxidation state of a series of proteins in the cytoplasm, where usually there is the general belief that being a reducing environment, all the proteins should be in the reduced state. And actually this is not true, because also the redox state of the protein is tightly controlled, and each individual protein has to be or in the reduced state or in the oxidized state, and this is controlled by specific proteins. And for example, this is a mitochondrial protein, which when is in mitochondria needs to be folded and oxidized because the disulfide bond stabilizes the structure, but in the cytoplasm need to be in the reduced state because it needs to be unstructured, and only as unstructured is able to enter the mitochondria and to go through the outer membrane of mitochondria. And however, if we overexpress this protein, this is a larger share of this is in the oxidized state. While if we co-express some specific redoxin, glutaredoxin or thyrodoxin, then we have the ratio between oxidize and reduce, which depends on the redox potential of the cytoplasm. And the same is true for other proteins, MIA40, and the same is true for SOD and as I mentioned, depending on the methyl state and depending on the presence of CCS. So this is just to show that also the redox state, which affected protein aggregation, is really tightly controlled by the presence of other cellular components. And this is another example of how in-cell NMR is very useful in understanding what is happening atomic level in proteins which are involved in pathological diseases with, as I said, atomic level upon, for example, oxidative stress. So this DJ1, that is also called PARC7, is linked in several neurological disease pathology, Parkinson and ALS. And this protein, you can see here, in this case, we have slightly increase in broadening, but still we have a spectrum which has very high resolution in the cell compared to the one in vitro. And this protein is sensitive to oxidation. It contains a number of cysteines, but which is the cysteine which get oxidized inside the cell? So then what we did, we treat cell with hydrogen peroxide and looking specifically in selectively labeled cysteine, we monitor that is cysteine 106, which get oxidized, the other remain completely in the reduced state. And then from the mass analysis, we identified that the oxidized species is SO2, while there were proposals that SO or SO3 are not formed at all. And that is, as I said again, this is a very good way to very precise atomic level information that we can have in the cellular environment. And these are while cells are still alive, because always at the end of the experiment, we check and that the cells are still alive. In any case, we would see spectral changes. So then we are totally sure that this is occurring while the cells are still alive. Another aspect we address is toxicity of cadmium. So cadmium has been proposed that cadmium is toxic and would displace zinc from SOD. And that is not true. Cadmium has low affinity, very low affinity for the zinc binding site. And specifically, if you look at this spectra, these are 1D proton NMR spectra, again collected on human living cells, then we can see that the spectrum SOD that is fully saturated with zinc, there is a little bit of zinc bound because in the growth medium there is always a little bit of zinc because it's needed for cell growth. So these are zinc added, and then you see that addition of cadmium doesn't change at all the spectra. So there is no effect on SOD. Still, there is an effect, and specifically there is oxidation, cysteine oxidation of SOD. So this is empty zinc without copper. The cysteine are still reduced state. And if there is extra cadmium, this induces oxidation. And so this is quite peculiar because it's not a redox metals. What is also peculiar, we see that addition of cadmium just to cells which are not overexpressing any protein induce very high overexpression of metallothionein at a level so high that we can detect it by in-cell NMR. So this was not induced by us, by transfection, but induced by cadmium. And so then we rationalize this behavior based on the fact that when just we add cadmium, cadmium release may remove zinc from the metallothionein which are in the cell, thus releasing zinc, and then zinc induce the production of further level of metallothionein, and this is why if we add the cadmium we have a further higher level of metallothionein. When there is SOD at a higher level, SOD sequester zinc, the zinc which is released by the addition of cadmium, obviously SOD transfection and cadmium, extra cadmium. Extra cadmium releases zinc, zinc is bound to sequestered by SOD, there is imbalance in the cell and this promotes oxidation. If we add extra zinc in addition to extra cadmium and transfection, then we reestablish all the process, because in this case, yes, there is extra SOD which sequestered the extra zinc and the cadmium is still bound to metallothionein. So just this is another example of how this type of analysis it can characterize this process in quite detailed in environmental, in a physiological environment. I would like to take the last few minutes to present a new type of applications of in-cell NMR that we have just developed in the last 1.5 years, 2 years, that is the use of in-cell NMR for the drug screening. And this is particularly relevant for several applications and for the drug optimization and design and optimization in the early steps. So what we tested this for a series of inhibitors to carbonic anhydrase, again in metalloenzymes. It contains zinc, and this enzyme is present in all our possible tissues, that we have several isoforms, many isoforms, and this enzyme contains zinc, which catalyze the transformation of CO2 to hydrogen carbonate, such a way that our blood doesn't have a bubble like champagne, but is solubilized as hydrogen carbonate. So this is an absolutely essential enzyme. Is a zinc enzyme. There is a OH bound to zinc, and this, if this is removed by molecules, then the enzyme is inhibited. And in some cases, in some diseases, this protein is overexpressed, and so then there are drugs already on the market in the drugstore that are the function is to inhibit this enzyme. And here is the example of, again, these are in-cell spectra, 1D spectra of the histidines bound to zinc. As they are coordinated to zinc, this proton of the NH proton of the histidine are shifted outside the magnetic envelope, and they do not exchange fast. They exchange slow enough that can be detected outside the magnetic envelope. And this has the advantages that we can exploit also simple 1D NMR spectra without the need of labeling. So we can label the protein. This is the spectra that is a quite reasonable spectra. High resolution now is compressed, but otherwise we can recognize all the signals. And this is a 32K molecular weight protein. So then still we can very good high resolution spectrum. But for this type of analysis, we can exploit also 1D spectra. And what I would like just to show you here is that these are some of these molecules we have screened and many other. You can see here that we can clearly see those molecules which bind and those which do not bind, that the spectrum is exactly the same as of the free control. And I want to stress molecule 1 and molecule 2. And this is molecule 1 and this is molecule 2. You see that the affinity constant is identical in solution for the two molecules, but one is binding and the other is not binding. And this is clear because in the I would say is not obvious, but is an important result, because one aspect of this affinity constant has been measured in the buffer, and this binding has been measured in the cell. So then there are molecules which do not enter the cell, other which are enter the cell but are sequestered by other components, other they are very slow diffusing in the cell. And so then obviously we can do a deep analysis. Now I don't think I have time anymore. Analysis of this as a function of dose dependence and time dependence, and we can analyze this behavior. But what was really, I think, an important point, that we found a striking correlation between the efficacy, the permeability values, and the efficacy in inhibit the enzyme and the doses of the two approved drugs. So they have similar affinity, but in vitro, but the dose, approved dose, one is 10 times more than the other. And then we found a clear correlation of what we can see in the cell. And these are very simple measurements. We can be done very early in the drug screening. Just a very simple, I skip this. We develop this bioreactor that is useful for, let's skip one and I will go to this drug. The bioreactor is very useful for keep the cell alive for a reasonable long period of time, because otherwise the cell closed in the NMR tube without oxygen and nutrients, they last for roughly 2 hours, while with the continuous flow of nutrients, we can keep them alive for 10th of hours. And then we can follow when the addition of the drug and how it diffuses. So this is what is the spectrum without drug, and then we can see the onset of the signals of the bound form. And here of these other inhibitors, we can see that the binding is much faster. It occurs much earlier in time, while this, it takes time to detect, to have the formation of the inhibited form. So this is really something that we can analyze in terms not only of the binding affinity and concentration, but also with respect to time. So I come to the end. I saw that Claudio, yes, my time is over. Just to show you a charm with our collection of NMR. Actually, this is older because it's 12 NMR spectrometer plus a relaxometer. And we have really the largest magnetic field range available, because we can do measurement at 0.01 megahertz up to 1,200 megahertz. And we have latest acquisition like cryo, electron microscopy, we have a pulse, EPR, and a number of other instruments. And here is the 1.2 gigahertz, and here is the 900 is here where we tested more than a year ago during the lockdown, the real severe lockdown. And we tested to the instrument. And with this, I want to thanks the people of the InCell NMR group, Enrico Lucinat, Letizia Barbieri, and Matteo Cremonini. We have also a magnet case outside the CERM, and I would like to thank you for your attention. Obviously, I'm ready to take any question. Thanks.
Thank you, Lucia. So I understand that we should look at the Q&A, but first there was a question in the chat from David Eliezer. Can I read the question? How does it work? Yes. So he says, beautiful work, Lucia. The results on the effect of SOD mutations in cell versus in vitro is really fascinating. Did you happen to look at DJ1 mutations? They have crystal structures and NMR spectra in vitro very similar to wild type, but I don't think they have been looked at in mammalian cells.
Yeah, so, no, yes, thanks for the question. No, the DJ1, we just limited to the behavior of the wild type. It might be interesting to expand to the mutants, because indeed the protein expressed well in human cells, and so then we can characterize. But no, unfortunately, we limited to those studies, to the oxidation behavior of a wild type.
We have a question from Franz Mulder. Great talk and very amazing work. My question is the following. We talk about in-cell NMR often as a way to avoid in vitro, such that the experiments can truly be translational. While we are aware that much of the NMR requires quite high levels of proteins present to be visible, what is your view on the use of the term in-cell in general? It would appear to matter a lot in these cases to be also specific about the types of cells that you are actually looking at and their cellular states. Second question, should we try to observe directly or quench, extract, and analyze?
Okay, there are many points. So this is a very long philosophical question. First of all, I mean, yes, it's true that in-cell is a generic name, and so then we should mention the type of cell and so on. As for any work we had in the experimental section, the conditions and so on. So otherwise it would be too long to, let's say, in Ecr cell or in ELA cell. Yeah, we can add, but I think it would be a little bit too cumbersome. Regarding the in vitro and moving to the in cell, I think that the two approaches would not exclude one the other. And the reason is because in some case, you need to have a really complete assignment, analysis, and I mean, let's say assignment, because, and that it has been done, assignment of a protein, but was a small protein, a bacterial protein, and was done in the cell with a lot of samples. Now, I think my approach is that we should use the fastest and most economical approach to get the most relevant answer. So then is the idea is that we have to look at what is happening to to your protein if are human protein in human cells. Then you could try to transfer that condition in vitro or in bacterial cells. And the idea is because if you are in vitro, with the assignment, you can be done in a single sample. If you have to do the assignment in the cell, you might need several samples. And definitely for human cells, the growth medium is incredibly expensive. And in the case of human cells, but any cells, the carbon-13, there are a lot of metabolites that you have the background of the metabolites. If you use carbon-13 in cell, the spectra became much more complex. So the idea is that we have the assignment in vitro, and if we have to optimize the spectra in such a way that is as close as possible to the one in the cell, and then we combine the two types of information. This is, I think, is the most efficient way. Then in the future, we might be, for example, with the bioreactor, we can keep a cell alive for longer period of time. We might exploit to do longer experiments. Still, the issue of the carbon-13 background is an issue. That is, and the cost of the growth medium for human cells. That is another label medium. I don't know if I have addressed your questions.
We have a question from Jason Chua, and the question is, has there been any look at the effects of riluzole or edaravone on SOD1? These are specific molecules, I guess.
These are, no. Personally, we haven't characterized any molecules on the specific case of SOD. We have characterized, as I said, the maturation and the effect of metal. That is, how to say, that is our bias in terms of aspect, but not on drugs or potential drugs. That it could be a good ideas. We have checked, we have analyzed epsilon, and epsilon that is, and we did with cisplatinum, again, because it affects the oxidation state of the cysteines. But we haven't analyzed other specific molecules. But it's a good idea. That is something we could check, because SOD is easy to produce in X cells. So.
We have a comment from Franz Mulder. He says the reason for asking the previous questions was that many proteins may not be seen due to interactions within the cellular milieu, which is of course.
Yeah, that is perfectly true, and indeed we see this in a number of cases. Still, we see that in cases when we see the protein is detected in the cell, in a number of cases, the behavior and the spectra, let's say this way, the spectra are different when they are inside the cell or when they are in the crude lysate. Not, I mean, just breaking the cell and taking everything, the spectra are different. So this indicates that the interaction pattern and so on is different when you have intact cell and when you have broken the cells. And this will, so obviously it's better to use lysate in case you don't detect the protein inside the cell, because we have weak interactions. In some cases, for some proteins, as we had an idea, which was in the interaction side, then we introduced mutations, which didn't affect the function of the protein, and then the signal became detectable. So this is another approach.
We have a question from Vipin. You mentioned that in the cell, copper is in the +1 state, while in vitro it is in the 2+ state. Can you comment on what factors could possibly lead to shift in the copper oxidation state?
Yeah, this is, I mean, in vitro is the oxidation state you add. I mean, we added as copper 2, but it can be obviously copper 1 as well. So that is, it was suggested, and that the NMR spectra demonstrated clearly that there is a protein responsible of the reduction of copper, and copper, once is uptake in the cell, is inserted in the reduced state. So that there is a specific protein which is responsible of the reduction of copper. So copper is inside the cell is present as copper 2 only when it's cycled in the redox reactions, like an SOD. In some state, obviously, copper is cycled between copper 1 and copper 2, and then copper 2 to copper 1, back to copper 1. The resting state is copper 1, but then obviously in the redox. But yes, a lot of metal ions, external are in a redox state, and when are internalized, are in a different state. So that are like iron, that is present as iron 2 because it's more soluble, but then there are cycle, and in some proteins bind iron 2 or iron 3. So then there is a specific enzyme for reduction of copper.
And as you have shown, I think copper is always transported and stored as copper 1 in cells.
Yes, yes.
It's only copper 2 when it's incorporated in the final target.
No,
when it's cycled,
in the rest, in the SOD.
But I mean, as you said, it can be copper 2 only when it performs its function in the proper protein. Otherwise, yes, it's always copper 1.
Yes. Is only in the cycle, in the redox cycle of the function, this copper is involved in redox reaction. And otherwise, resting state, we can see clearly from the NMR spectra and the 1D NMR spectra. And I mean, we have spectra of the oxidized and reduced. And also from the ratio of the signals is clear, is let's say 95% is in the reduced state.
So I think your 10 minutes questions are over. I don't see any more questions, so we are almost perfect in time, and I leave the floor to the next chair and the next speaker.
Thank you.
Great. Thank you very much. I'll introduce our next speaker, who is David Eliza from University of Cornell Medical School in New York City. David is actually a native of Michigan and received both his bachelor's and master's degrees in computational science from the University of Michigan, though I think that predated Rams's arrival in Ann Arbor. After completing his master's in computational science, he went to Stanford, where he completed a PhD in physics with Seb Doniak, and David did really pioneering work on the small angle X-ray scattering of proteins. Of course, a widely applied tool these days to intrinsically disordered proteins and other systems. But David's work in the 1990s was really pioneering in that regard. And I think one thing that stands out is their study, which was the first time-resolved study using SACs to look at protein folding intermediates, and is still one of the most cited papers in that area. After completing his PhD with Seb, David moved down the California coast to La Jolla and did a postdoctoral research appointment with Peter Wright and Jane Dyson at Scripps, where he worked on molten globule intermediates, structure and dynamics of protein folding intermediates, and branched out to really extensive NMR work. It was in 1998 that David began his, 1999, I apologize, began his independent career at Cornell, where he began to work on NMR of alpha-synuclein. And it seems fairly obvious these days that we would like to use NMR to look at intrinsically disordered proteins, systems that aggregate. But when David started, he was really the pioneer in this field. Those who did NMR didn't think it was that interesting because it didn't have a structure. Those who worked on the biology of this thought it wasn't very interesting. And so David deserves a lot of credit for really pushing that field forward. David is known for some other things. We could regard perhaps his work on the small angle X-ray scattering and his initial work on folded proteins and as first ascents. But David has also had a few other ascents. And I think this is a nice example that you don't always have to be the first person in science to do something important. This is a picture of David, which I coerced him into letting me show. And what he's doing here is repeating an experiment that was first performed in 1957. This is the ascent of Half Dome in Yosemite National Park. It's an 1,100 meter high sheer rock face, which is one of the classic rock climbs in the world. David's hobby was rock climbing when he was a postdoc and graduate student. Not only did he do an ascent of Half Dome, he did an ascent of the other famous face, El Capitan, and those are probably the two most challenging rock climbs in the world. So rather more impressive hobby than stamp collecting or bird watching. And I'll stop embarrassing David and just mention a little bit about his more recent career. David continues to do really important work on alpha-synuclein, and he also works on the snare regulatory protein complexin. He works on tau and the role of post-translational modifications in tau aggregation. David is the recipient of a number of awards. He's won the National Award of the Parkinson's Foundation. He's a recipient of a U.S. National Institutes of Health Merit Award, as well as the Irma Hirsch Award. David has also been a superb colleague to the field. He's served on an enormous number of NIH, NSF, MRC, and Wellcome Trust grant review panel, serves on the editorial boards of a number of journals, and has organized and chaired a number of conferences. And I believe that today, David is going to tell us about some of his recent work on alpha-synuclein. So David, I'll turn it over to you and look forward to your talk.
Thank you, Dan. Let me share my screen, hopefully without any technical issues before I start anything else. Okay, hopefully you can see the title slide. And I want to start by thanking the organizers for giving me the opportunity to share some of our work with you. It's been a challenging year, I'm sure, for all of us. And so it's nice to be able to focus on science and forget about all the other stuff that's going on. I also want to warn you that there may be some background noise on a Saturday morning at home. It's not always quiet here. I've been trapped in this house with 3 small creatures for the past year. And so I've told them not to bother me, but I can't guarantee that we won't have interruptions. And I also want to thank the organizers, not just for inviting me, but for holding this series. I haven't been able to attend many of the lectures in person, again, because the timing is a bit difficult with young children, but I have enjoyed a lot of them, the ones I couldn't attend in person, on the YouTube channel. And it's really an amazing collection of speakers and of research, and I've really enjoyed it. And I'll refer to my enjoyment of one particular recent seminar in a little bit. So thanks, Dan, also for the very, very kind, overly kind introduction, but also for the rock climbing bio, because it's good to have sometimes pursuits that clear your mind and let your subconscious work on things. And I would say rock climbing, which is actually very popular with scientists and physicists in general, falls into that category. So with that, I want to start my talk first with the acknowledgements, because I know the time is limited and I have a lot of slides and I don't know if I'll get to the end or not. So I want to acknowledge the hero of today's talk, who is a graduate student in my lab, who's just finishing up, just defended his dissertation and is moving on to postdoctoral work. He hasn't quite decided where yet. And so this is Tapo Das here, shown on the left. And he's the main player in all the data that I'll show you today, the structural data that was collected in my lab. But the story I'll tell you today is also primarily a fantastic collaboration with a laboratory in the mothership campus, as we call it, the Cornell campus on Ithaca. Our medical school campus is in New York City. The main campus is in Ithaca. And that is the lab of Barbara Baird, David Holoka, and their postdoc, Marcus Wilkes, started this work in their lab, and it was continued by Raj Ramazani. And this is where most of his cellular assays were performed, although Tapo did travel up to Ithaca and do some cellular assays as well. And then I'll talk a little bit, if I have time, about a work that's a collaboration with Peter Chung while he was a postdoc in Kai-Yi Lee's lab in Chicago on synapsin. He now has an independent lab he just started at USC. And I'll also briefly mention some data from a long-standing collaboration we've had with Halal Lashuel, and some older data that was a collaboration with Watt Webb and his graduate student, Valerie Anderson. And of course, we couldn't do any of it without the funding, and also the New York Structural Biology Center, where we have our shared high field instruments, is critical to our efforts. And we often do ESR in collaboration with Jack Fried, although at a search center in Ithaca, although I won't talk about that data much today. So I think most of the people in this audience, this being a meeting focused on proteinopathies, are familiar with alpha-synuclein. So I don't want to spend a lot of time on this slide, but I will say that the reason most people are interested in alpha-synuclein is summarized in this slide, and that is because it's linked via pathology, Lewy bodies, to Parkinson's disease, where it's found in an aggregated form, and it's also linked genetically to Parkinson's disease via specific mutations and or duplications or triplications, increases in the level of the protein. And most people are convinced that this is what's interesting about this protein. And what most people are not so interested in necessarily is, what does this protein actually do? Why is it there in the first place? So most of what I'll tell you about today is actually our attempt to understand why this protein is around in the first place and what it might do. And so it's called synuclein because its localization, when it was first observed in electric eel, was primarily presynaptic nerve terminals. There was also a nuclear localization, which remains controversial and even more poorly understood. But even at the presynaptic nerve terminal, where everybody agrees there is a lot of synuclein, we don't really understand what it does, but somehow it seems to influence the synaptic vesicle cycle and synaptic vesicle release. And these images that were collected many years ago, not in my lab, I think this is David Sulzer paper, show that synuclein stained here co-localizes strongly with the synaptic vesicle marker. This is a pump that sits permanently on the synaptic vesicle membrane. It has it's a transmembrane protein. And so it's a strong marker for presynaptic nerve terminals. And synuclein co-localizes nicely with VGLUT1. But if you look, you can also see that there's quite a bit of staining or fluorescence in the processes themselves. And so that tells you that synuclein is not permanently parked on synaptic vesicles, but it's rather a peripheral membrane protein that interacts transiently with synaptic vesicles. Already you can tell that from these data. So as Dan kindly mentioned, we've been studying this protein for many, many years. Started my lab in 1999, and I actually started working on synuclein a little bit while I was still at Scripps. And this was all really began by Peter Lansbury, who came to Scripps and talked about this weird protein that didn't seem to have structure and yet was functional and only was starting to be associated with disease back then. And I thought, oh, this might be an interesting protein to study. And Peter Lansbury said, I've tried to get every NMR spectroscopist, within a 100-mile radius of Harvard to look at this, and nobody's interested. So we were lucky that that at the time we had this to ourselves for a little while, not for very long. So we and many others have spent a lot of time, and I just show some of the data here to show that this cartoon is not just a figment of my imagination, but is actually based on data, but I won't go through in detail. And the picture that has emerged over the years is that alpha-synuclein is very highly disordered when it's isolated in solution. We believe this is the case in the cytosol of cells as well. There was some controversy about this in recent years, but I think this still stands. under most circumstances. However, it does interact with membrane surfaces, and especially things that look like synaptic vesicles, as much as we can make them look like synaptic vesicles in vitro. And when it does that, 100 out of 140 residues adopt this long alpha helical structure that we call the extended helix state. But if you look at the protein in the presence of smaller lysophospholipid particles or detergent micelles, the single long helix breaks into two shorter helices connected by a linker, and we call this the broken helix state. So we've been trying to connect this protein, these structural snapshots, if you will, to the function of this protein for some time. And it occurred to us that we needed to know something about what people have seen this protein do in cells. And so there was already data for some years indicating that alpha-synuclein influenced synaptic vesicle release in cells. And so we took this cartoon vignette and we superposed it on a synaptic vesicle that's ready to fuse with the plasma membrane. And this has become for many years now our sort of working model of what alpha-synuclein might actually do in the cells. And the idea is that the extended helix state and the free state are in, exchange. This is a weak affinity interaction in vitro. It's also a weak interaction in vivo. As I showed you, synuclein is not permanently parked on the vesicle. It exchanges back and forth. And only when the vesicle becomes docked is there a high affinity binding site created between the two membranes. And we postulated that that would be bound by the broken helix state with the protein bridging between the two membranes, and that this would allow the protein to somehow influence the exocytosis process, which is mediated, of course, by proteins that I don't show here, the snare proteins, the monk proteins, complexin as well. And this membrane bridging model didn't gain traction for a lot of years, but in more recent years, Giuliana Fusco and her group have also, I think, sort of adopted this idea that this may be an important type of interaction for alpha-synuclein, and they've done some very nice work in that area as well. So why would one be interested in studying the normal function of this protein when really it's sort of amyloid fibril formation that's likely links it to Parkinson's disease? So that's a bit of a philosophical question. I want to, just answer it in a few brief answers to that question. One, of course, is basic curiosity. Two is that attempts to treat aggregates of various amyloid diseases so far have been largely unfruitful. And so Christian a few weeks ago told you guys about the Biogen studies, the antibody studies. None of those have really panned out. It may be that the FDA will approve aducanumab, and if so, in my opinion, that will sort of be running a clinical trial on the population to see whether it works. But the only drug really that's approved at this point to treat an amyloid disease is a compound created by Jeff Kelly that targets transthyretin, and it doesn't target an oligomer, it doesn't target the end of a fibril, it doesn't clear transthyretin deposits from human brain or organ. What it does is it targets the native state of transthyretin and traps it in a state where it cannot enter the aggregation pathway to begin with. So we've always in the back of our mind thought, well, maybe there's somewhere out there a functional state of alpha-synuclein that might be a good drug target, and a better drug target, for example, than this intrinsically disordered state, which people have tried with varying success to drug, but so far certainly nothing in the clinic. And the final thing is that we don't really understand why and where synuclein aggregates in the cell, and I think that's going to be very important ultimately in our ability to target what synuclein does. Okay, and I will support that last assertion by saying that this model is useful to us. Of course, it's a model. This is very hypothetical still. It's been useful to us. And so one thing that we did early on was think about what could govern the transition between these two states of the protein. And we knew that oftentimes conformational transitions in proteins are regulated by post-translational modifications. And I don't show it here, but at the end of this first helix, there's a tyrosine residue, tyrosine 39, which is the only aromatic residue within the lipid membrane binding domain other than a phenylalanine to very N terminus and a phenylalanine to very C terminus. And we always thought that this tyrosine kind of stuck out like a sore thumb and postulated very early on that it might be a phosphorylation site. And we also thought perhaps that phosphorylation site would somehow regulate this membrane binding process or the conversion between these two states. So eventually with Halal Lashuel, who wasn't scared of doing things like raising antibodies to a phosphorylated peptide corresponding to alpha-synuclein, we showed that in fact tyrosine 39 alpha-synuclein is phosphorylated. It's phosphorylated by c-Abl, which was at that time shown to have links to Parkinson's disease. And when you phosphorylate tyrosine 39, what you do is you kick off this second part of this membrane-bound helix off the membrane surface. And we thought that this may be a functional, consequence that allows this part of the protein to then go seek out and bind another membrane like the plasma membrane. So these are some of the NMR data showing the effect on membrane binding. There's a Parkinson's disease mutation that was discovered around this time that does the same thing. We had shown earlier that when you create such states that are partly helical, they are potent drivers of aggregation. And so one of the things that came out of this model that led to the study and the discovery of this phosphorylation site is the idea that not only is membrane binding functional for alpha-synuclein, but membrane binding may also, in some circumstances, be pathological. In particular, if you create states on the membrane where part of the protein is helical and bound to the membrane, but part of it is not, especially this NAC domain here, then that may cause the helical regions to come together, associate with each other, and align the NAC regions in a way that might drive fibril formation. So I was really very happy to see Christian's and Life's talk a few weeks ago, where Life is basically looking at something that we think is almost exactly this, where they see membrane-bound conformations of the protein, where the N-terminus is still in helical conformation, but this part of the protein is progressively forming amyloid fibrils. And I think that's really cool and supports the idea that membrane-induced fibril formation may be important. And of course, the idea that helical formations contribute to amyloid is not unique to synuclein. It exists for many amyloid proteins, A beta, IAPP, and Dan has contributed to this, and many others as well to this idea. So I think it's still an interesting and valid idea. But I won't talk today about aggregation. I'm going to talk about this part, the functional aspect of alpha-synuclein. So it was nice to have this model. It led us to discover phosphorylation of tyrosine 39 and its putative functional effects on the protein. But we really wanted to do more than that. We wanted to do structure function on this functional state of the protein. And structure we could do in vitro, but function we really didn't have a good assay for function. So we reached out to Barbara Baird at Cornell. We reached out to many people, and it was hard to find somebody who was willing to take this on. But Barbara Baird, who works on cells that are professional secretory cells that are sort of a model for immune cells, and asked her if her lab would be willing to join us in setting up a functional assay for alpha-synuclein. And they said yes. And the assay that we used is the fluorine assay that's been looked at originally for release of synaptic vesicles and neurons, but can look at many secretory processes. And the idea is that you have a pH-sensitive fluorophore, a GFP variant inside the lumen of vesicles, secretory vesicles or synaptic vesicles. The fluorescence is largely quenched at acid pH. These vesicles are usually acidified, but when the vesicle fuses with the extracellular medium, the pH is neutralized and you get a buildup of fluorescence on the cell surface. And so this is a nice assay for monitoring vesicle release. And Barbara's lab applied this assay in model cells. These are RBL2H3 cells, so they're not neurons, but they're nice, big cells that are easy to work with. And when you stimulate these cells, these are the control cells here, no synuclein present. Over time, you see a buildup of fluorescence on the membrane surface, and that is telling you that the vesicles are being released as you would expect. If, however, you transfect a little bit of synuclein into these cells and repeat the experiment, what you see is that release is very potently inhibited by alpha-synuclein. And this wasn't something new or startling. People have already shown this to be the case in neurons, that alpha-synuclein was capable of inhibiting vesicle release, synaptic vesicle release, and that's in part what led to the model that I showed you. But now we had the assay in our hands where we could now go back and mess with the protein, look at what that does structurally, and then ask what that does functionally, which is what we wanted to do. So we were very pleased at having this assay, and we set out to do structure function. So this work was published a little bit before the pandemic, so I'm going to go through it pretty quickly. What I want to remind people is that we are actually looking at what happens in vitro, and we are assuming that when we change things in vitro, that also leads to changes in the cells. And we assume that when we perturb this state of the protein, we're changing this binding mode in the cells, and that when we perturb this state of the protein in vitro, the consequence in cells that we're perturbing this binding mode. So that's the logic behind all these experiments. That is an assumption. We haven't in any way shown that to be the case, but that's the logic behind this. So I just like to be upfront about it. So we made mutations that altered these states of the protein. We asked what happens. And again, I'm summarizing here. You can see the paper for the details. We found that mutations that perturb this binding mode, especially that perturb the binding of this second helix to the membrane, eliminated the ability of alpha-synuclein to inhibit vesicle release in our cells. And so this, again, wasn't too surprising, but it provided some support for the idea that this membrane bridging model is not entirely fantasy, and in fact underlies the ability of the protein to inhibit vesicle release. And that was nice. And we're pursuing this line further because we only have a handful of mutations and we want to really explore, what's important. We already have some additional evidence that the linker actually plays an active role, and there's a lot still to do there. But we also had a bit of a surprise, which I'll tell you about next. So I told you that people are sort of skeptical of, why somebody would study alpha-synuclein function. And my colleagues came to me and said, well, this is all very nice. I know you're interested in it, but nobody else is interested in it. And in particular, nobody's going to fund you to do this work. So you better study synuclein aggregation because that's what it's all about. And so we said, well, what can we do? I guess we can try to add a lot more protein to these cells, and then maybe we'll see aggregation, and then we can do structure function of aggregation, which might also be interesting. What's important for initiating aggregation in cells. We could ask interesting questions. So we added a lot more protein to these cells, and we never saw any aggregation. So we didn't see aggregation with a little bit of protein. We didn't see aggregation with a lot of protein. We looked by cross-linking. We looked by Western blots. We looked by localization. And we really never saw convincing evidence that we have aggregation going on in these cells. And it's a little bit of a dirty secret, I think, both for tau and alpha-synuclein, that these proteins are actually exceedingly soluble, both in vitro, but also in cells. And it's not so easy to get them to aggregate. It's quite challenging. And so in vitro, you have to add cofactors or shake or raise the temperature. And in vivo, in cells, usually what you have to do is add some sort of a seed, whether PFFs or something else. So we didn't see aggregation. We have no way to study aggregation in these cells at present, but we had, highly cells transfected with a lot of alpha-synuclein. We said, what can we do with them? We said, well, let's run our assay and see what happens. If a little bit of alpha-synuclein inhibits vesicular release, a lot of alpha-synuclein should really kill vesicular release, right? And here we got a surprise. And so the student came back and said, well, no, when I transfect in a lot more alpha-synuclein, I actually see earlier release and more release. Okay, there's more fluorescence building up on the membrane. It starts earlier. And I said, of course, go back and do the experiment again. You messed it up. The cells are bad, make a new batch, et cetera. Especially because I know very little about cell culture and cell biology. But Barbara looked at this over carefully, and this is very reproducible. We did this many times, and it always came back the same. So then we had to face reality. We said, okay, this is the data. It appears that a lot of alpha-synuclein can actually potentiate release of vesicles. How could this be? So then we said, okay, well, we have, the ability now. We have an assay that for potentiation of vesicle release, and of course, we have our structural studies in vitro. Let's do structure function. So again, we looked at various mutations and their effects on the different membrane binding modes of alpha-synuclein, and we found that mutations that affect this binding mode of the protein, but not this binding mode of the protein, actually eliminate the ability of alpha-synuclein to potentiate release at high levels. So this was comforting in the sense that it told us that these were two different functions of the protein. But now we have to go back and think, well, what does this mean? That means that this mode of the protein now also has a function that we didn't know about before. And what could that function be? And how does it lead to potentiation of vesicular release? So we had to go back to the literature again and ask, is there evidence in the literature that alpha-synuclein somehow influences vesicles that are not bound to the plasma membrane, not docked to the plasma membrane and ready to fuse, but in fact may not be at the plasma membrane at all, because for this binding mode, you don't need another membrane. So we went back and looked at the literature. And in fact, there were two papers that had very interesting results that related to effects of alpha-synuclein on vesicles that are not docked at the plasma membrane. So first, there was a study by Namani et al., which was looking at overexpression of alpha-synuclein in cells. And what they showed is that when you have a lot of alpha-synuclein in cells, it actually changes the ultrastructure of presynaptic nerve terminals. So on the left here, we have wild-type nerve terminals. And what you see is that the synaptic vesicles are clustered, and they're clustered near to what's called the active zone. This is where they are going to fuse so that the postsynaptic neuron can detect the fusion event, or the release of neurotransmitter. And so you see these clusters in the wild-type neurons, but in the neurons that have synuclein overexpression, these vesicle clusters disappear, and the synaptic vesicles are instead dispersed throughout the presynaptic nerve terminal. Now, Sriranga Chandra at Yale did the opposite sort of experiment where they eliminated synuclein entirely from entire mice. So they looked at triple knockout mice, alpha, beta, and gamma knockout mice. And in short, what they saw compared to wild-type mice, where the synaptic vesicles are normally clustered, in these triple knockout mice, the synaptic vesicles were very, very densely and tightly clustered. And this was true both in cultured neurons and in slices from the brains. So the message that came out of this is that synuclein does affect vesicles that are not docked at the plasma membrane, and a little bit of synuclein keeps the vesicles in their normal clusters. If you don't have any synuclein, they're very tightly clustered. And if you have a lot of synuclein, then they're not clustered at all. So the more synuclein there is, the more loosely the synaptic vesicle clusters are organized. And we thought this was interesting. Now, the issue was that, unfortunately, we were not working in neurons. We were working in RBL2H3 cells. And so we first had to ask, where do our vesicles that we see fuse with the plasma membrane, where do they come from? And the vesicles we're looking at are recycling endosomes. And so this is their trafficking cycle. And the place they come from before they bud off and go to the plasma membrane is an organelle called the endocytic recycling compartment, or ERC. So this is sort of the equivalent of the synaptic vesicle clusters in neurons for us. And we said, well, if synuclein appears to affect synaptic vesicle clustering, maybe it does something to the ERC in our cells, and could we look at that? So there's a well-known marker for the ERC, which is called RAB11. And so we made RAB11 MRFP fusions, and we looked at those in our cells and asked, what does expression of high levels of alpha-synuclein do to the ERC? So this is what ERC staining looks like in a cell that's not transfected with alpha-synuclein, or is transfected with low levels of alpha-synuclein where there's no potentiation of release. And the ERC is a perinuclear organelle, so most of the staining is inside the cell, and there's some staining at the periphery of the cell. And we believe that this is vesicles that have trafficked to the plasma membrane and are the equivalent of docked synaptic vesicles that are ready to fuse. If you transfect in high levels of alpha-synuclein, what you see is a redistribution of the fluorescence. You have less fluorescence inside the cell and more fluorescence at the periphery. And so we believe that something similar is going on here to what we see in neurons, that vesicles are being dispersed from intracellular stores to the periphery of the cell, and that this is likely responsible for the additional release that we see at high levels of alpha-synuclein. And that's supported by the fact that one of the mutations that eliminates potentiation of release also eliminates this redistribution effect as quantified here. So all of this data together led us to this model, which is that for both synaptic vesicles in neurons and recycling endosomes in our RBL2H3 cells, alpha-synuclein at low levels or at any level interacts with docked vesicles, we believe via this membrane bridging mode, and in doing so exerts an inhibitory effect on the release of these vesicles. The mechanism for that is something that we still don't understand. At higher expression levels, alpha-synuclein engages vesicles that are not docked, and in neurons, it disperses vesicles from vesicle clusters, and in the ERC, it disperses vesicles, in RBL2H3 cells, it disperses vesicles from the ERC. And one of the things that we had to reconcile in this model is the fact that in our cells, this dispersion of vesicles from intracellular stores leads to enhanced release, whereas in synaptic vesicles in neurons, when you have more alpha-synuclein, people have looked at overexpression of alpha-synuclein in neurons and they don't see enhanced release. And so this was a contradiction we had to think about and explain. And our explanation for this is pretty simple, and it comes from the fact that these cells are not specialized cells. So in these cells, if vesicles leave the ERC, no matter where they go, they end up on the plasma membrane. And when you stimulate the cells, these vesicles can fuse anywhere in the plasma membrane, so they will all fuse. And so you'll get a potentiation of release by removing them from the inside and moving them to the outside. Now, in neurons, it's a completely different story. The vesicles are clustered near to the active zone because this is the only place they can fuse. They can't fuse anywhere else on the cell. So if you disperse vesicles from synaptic vesicle clusters in neurons, as overexpression of alpha-synuclein does, you don't get enhanced release because the vesicles all go to the wrong place where they're not able to fuse at all. And so this is, we believe, the resolution of this different effect of high levels of alpha-synuclein in our cells and in neurons. But fundamentally, we think synuclein is doing the same thing. Now, the next question we wanted to address, and I do still have a little bit of time, is how is it that synuclein might be dispersing vesicles from synaptic vesicle clusters or from the ERC in either cell type? So we started with thinking about neurons. And the first question we had to ask, well, how are synaptic vesicle clusters organized in the first place? So it's been known for many years that at least one protein that's critical for synaptic vesicle clustering in neurons is a protein called synapsin. And it was thought for many years that synapsin cross-links vesicles mechanically by binding to multiple vesicles at multiple sites. But this model never really held up to testing. And so it's been somewhat of a mystery since the protein was discovered in Paul Greengard's lab. How is it that synapsin actually cross-links synaptic vesicles? And this is old data showing that if you knock synapsin out of neurons, these are wild-type neurons, you have nice clusters, okay? And if you knock it out, the vesicles are dispersed. So these effect here looks a lot like synuclein overexpression. So a couple of years ago, Pietro Di Camilli published a paper proposing that synaptic vesicle clusters may be mediated by liquid-liquid phase separation, which is quite a craze that I'm sure you've all heard about, so I won't spend time introducing you to what that is. I'll just go over quickly Pietro's data. What he showed is that synapsin in vitro can form droplets, condensates, and that is not unusual at this point. Many proteins do this, but he also showed that if you add vesicles to these samples, then the vesicles labeled by a different dye here co-localize inside the condensates, so that these synapsin condensates recruit synaptic vesicles. And he proposed that this liquid-liquid phase separation of synapsin is the mechanism by which synaptic vesicles are actually clustered in neurons. And we thought this was a very interesting idea. And it occurred to us then to ask the question, is it possible that synuclein somehow interferes with these synaptic vesicle clusters, either by dissolving the synapsin droplets or simply by preventing the vesicles from being trapped inside the droplets? And so Topo decided to do some experiments to test this out. So the first thing he did was use fluorescently labeled synapsin to see if we could get droplet formation to reproduce what Pietro had seen. And we could. He got droplets both by DIC and by fluorescence microscopy. And then he added fluorescently labeled vesicles to these samples. And so these two pictures go together. Here we're looking at the synapsin EGFP channel. So these are the synapsin droplets. And here we're looking at the fluorescently labeled vesicles. And we see that the vesicles co-localize very nicely with the synapsin droplets, as Pietro's lab had shown. And now we could ask what happens when we add alpha-synuclein. So first, we looked in the absence of vesicles to see whether synuclein somehow dissolves the synapsin droplets. So this is looking at synapsin droplets in the absence of synuclein. And this is the same condition now with excess of synuclein. And we see that synapsin still forms very nicely droplets on the slide. So synuclein does not appear to somehow interfere with droplet formation. So then we asked what happens to vesicles getting trapped in the droplets. So this is the situation with no alpha-synuclein. Here we see synaptin droplets. Here we see fluorescently labeled vesicles. And you can see the vesicles localized to the droplets very strongly. And if we add alpha-synuclein now in excess, we see that unlike previously, there is still some localization of the vesicle fluorescence to the droplets, but it's much, much less. And we can quantify this, which Topo did. And so this shows the extent of vesicle co-localization with protein synapsin droplets in the absence of synuclein and in the presence of a slight excess of alpha-synuclein. And you can see there's a very significant decrease. So, and there's also a recent paper by Milovanovic showing similar data as well. So it appears that alpha-synuclein can inhibit vesicle release, but it can also enhance vesicle release. Enhancement is mediated by dispersing vesicles from intracellular stores, and that process may result from the release of vesicles from liquid-liquid phase separated condensates that are mediated in the case of neurons by synapsin. But of course, our experiments were not done with neurons. Our experiments were done with RBL2H3 cells, and so there aren't synapsin-mediated vesicle clusters. What we have is an ERC. So we don't know yet, but one implication of our experiments is actually the question, is it possible that the ERC itself, it doesn't actually look like this, is not a tubular reticular organelle that looks like the Golgi or like the ER, but might actually be some collection of membranes that's organized by liquid-liquid phase separation. And when I first suggested this to my colleagues, they laughed at me and they said, that seems very far-fetched and unlikely. But I was happy to point out to them an opinion piece, not containing any data, but a guy who has some credentials anyway, where he's proposed that even the Golgi itself might be organized by liquid-liquid phase separation. And I think this is a very interesting topic that remains an area of exploration, and we're looking into it a bit ourselves. So I think I still have 5 minutes left, I hope. I'm going to go on for 5 more minutes unless somebody stops me. So we've moved a little bit further with these structure function studies. And so I want to tell you a little bit about that work. So Topo decided that if in fact we believe that the broken helix state and the extended helix state have 2 different functions, it would be interesting if we could confine the protein to one of the 2 states and ask, or to either of the 2 states and ask, what are the functional consequences then? So he and a previous student in the lab made mutants that converted this linker region here either to 4 glycines, which would be very difficult to propagate an alpha helix through, or to helix promoting residues, which should stabilize the extended helix state. And he did NMR to prove that these mutations have the correct structural consequences. So in green, these are alpha carbon secondary shifts. In green is the 4G mutant. It has a very strong break between the 2 helices in the micelle bound state. In blue is this what we call 3AE mutant, and it has helix that propagates through this break that you see in the wild type protein. And then we looked at vesicle binding by these 2 mutants. And the 3AE mutant in purple binds very similar to the wild type. These are now intensity ratio plots in the presence of vesicles. So the wild type and the 3AE have this extended helix binding mode. But for the 4G mutant in orange, you see there's binding up to position 36 or so. And then there's a step function in binding where you lose binding past position 36. So only helix 1 here is able to bind to the vesicle surface. And helix 2 does not bind very well because of this interruption in the helical structure. So that was nice. The mutations succeeded structurally, and then we wanted to ask, what do they do functionally? So we collected functional data, Topo did himself, asking how do these mutations affect exocytosis at low expression where there should be inhibition, or at high expression where there should be potentiation. And he saw that compared to the wild type, the 3AE mutant did not inhibit as well, consistent with the idea that it doesn't really form the broken helix state very well. The 4G mutant didn't inhibit at all. This is a data point that we're still struggling to understand, but that implies that something about the linker is actually important for inhibition as well. This mutant can form the broken helix state, but it's not the right broken helix state. Something about it doesn't work, so this is a mystery still. At high expression levels, he saw that the 3AE mutant enhances release even better than the wild type, consistent with the idea that it promotes the extended helix state. And the 4G mutant actually also potentiated release to some extent, which at the beginning we were sort of mystified by. But we think we have an explanation for this now. One of the things Topo realized is that it's important to actually understand if our model is that binding to vesicles in the extended helix state is important for potentiation, we need to know the actual affinities of these different mutants for isolated vesicles. So to measure those affinities, NMR is not particularly good at that. You have to be working well above the relevant KDs. He resorted to tryptophan fluorescence. He put a fluorophore in the form of a tryptophan in position 4, replacing a phenylalanine, and collected fluorescence spectra. And you could fit those spectra as a linear combination of the free state spectra and the bound state spectra to get a fraction bound. And you could then do titrations and extract the KD. So these are the data that he got from his fluorescence titrations over many log units of lipid concentrations. How you fit these data is something that I'm not going to go into right now because of shortage of time, but we used the model that was initially proposed by Jennifer Lee, and you can combine this with data from the NMR titrations to determine sort of how many lipids do you need per binding site, and then how much of affinity is there, what's the KD once you have enough lipids to form a binding site. And doing this, Topo was able to generate a curve that correlates down here. Again, I don't have time to go through the details of the data. The correlates, you can't see the bottom, sorry, correlates the effects of exocytosis with affinity for lipid vesicles. And what he saw is that mutants or wild type, which have a relatively high affinity for vesicles, are able to potentiate exocytosis very well. And as you decrease affinity, you decrease the ability to potentiate, and at some point you lose it entirely with these 2 mutants. But that explained why this 4G mutant, which only binds via the N-terminal helix 1, is still able to potentiate exocytosis by alpha-synuclein. And it really directly supports the idea that if the protein is able to bind to the vesicle surface, that in and of itself will provide some ability to potentiate exocytosis, presumably by releasing vesicles from intracellular stores. So this is the model that comes out of this. Alpha-synuclein is able to bind to vesicles that are clustered, either in the form of an ERC or synaptic vesicles. It disperses them, okay? So this effect will stimulate, it'll lead to a higher percentage of vesicles that are available for fusing. But once the vesicles are actually docked and fused, alpha-synuclein sort of gets in its own way. It actually has an inhibitory effect via this membrane bridging mode. We don't understand exactly how this works, and we're still trying to understand that in detail. This idea, this here may be related to liquid-liquid phase separation. We're still pursuing that. And this likely has to do with some effect on the snare machinery itself that mediates the membrane fusion. So I'll stop here. We have some more data on the linker mutants that is actually quite interesting, but I think I'm already at the end of my time. So thank you very much. And if there are questions, I'll be happy to take them.
Great. Thank you very much, David, for our really stimulating talk. So we have a number of questions. We can do this 2 ways. I can either read them out, or if you want to bypass the middleman, you can just look at the Q&A thing directly. So let me know how you'd like to do it. And for the rest of the audience, if you happen to have some questions from our first talk, please feel free to pose those, but why don't we wait till we get through this for David, and then if you have any more questions or Lucia, feel free to post them in the Q&A.
Let's see, I guess I can look at them. So Christian, hi Christian, asked a question. I suppose I should read it both to myself and to you. Hi David, there are two papers by Bourret and Sudoff where they say that small oligomers enhance release while large oligomers, maybe at high concentration, block release. And there's also a paper by Yeon-Kyun Shin. Yes, I know that paper as well. So maybe in your high expression cells, you have the Bourret situation. Yeah, this is, again, the problem with cells that it's much harder to tell what's going on unless you're Lucia, perhaps, using NMR than when you're working in vitro. Again, we've tried this cross-linking approach that Dennis Selko used to propose the tetrameric state, which, again, I think his data was actually more consistent with oligomeric species than with well-defined tetramers. And we don't see really any cross-linking to suggest that we have even oligomers, much less fibrils. So it's not impossible, but so far we don't really see that. And I have my own opinions about the idea that synuclein clusters on synaptic vesicle. These data were based on FRET. The synaptic vesicle is pretty small to begin with, so there's going to be potentially some FRET even if you don't have real protein association. So I think this is an interesting area to look forward to. But what you really need, I think, are mutations that would would eliminate oligomer formation, and then you would ask, do they eliminate the effect? And so far, those don't exist. While we do have pretty clearly mutations that decrease the affinity for naked vesicles, and those do correlate very nicely with a functional effect. So I think that's my answer for that. All right, Alfonso. Hi, Alfonso. Ciao. Thanks for the great talk. Recently, there's been evidence that alpha-synuclein is also forming a liquid-liquid phase separation that condensates by itself. What is your view on it in view of your model alpha-synuclein interferes with LPS of synaptin-1? Yeah, so this is, of course, an excellent question. I have to say that we've kind of stayed away from liquid-liquid phase separation because my view is that it's kind of following a little bit the trajectory of amyloid. And those of you who are familiar with Chris's work, which you are and Dan is, and probably all of you are, know that Chris was instrumental in showing that essentially any protein can form amyloid. I was working on apomyoglobin in Peter Wright's lab, and we would routinely make samples that would gel in the NMR tube, and we would throw them away. And so I really startled when Chris Dobson published a paper showing that apomyoglobin can form amyloid fibrils, and that all those gels we've been throwing away were actually a science paper. So it was very, very cool. But a lesson for the graduate students out there: don't throw your samples away when they do something funny. So, I think a lot of things undergo liquid-liquid phase separation. Probably almost, I think, there are groups in India, I forget right now, who've sort of gone through many proteins and shown they can undergo LPS. So the question becomes, when is interesting? So I think for FUS and for TDP43, there's really good reason to believe that LLPS, the condensates, are related to aggregation and disease. I think the case for synuclein and for tau, I'll stick my neck out there a little bit, I think is less compelling. So synuclein, as far as I know, is not involved in any sort of punctal organelle. It's not involved in P-bodies or RNA bodies of any sort. So we've not really been terrifically fond of the idea that synuclein liquid-liquid phase separation on its own is very physiologically relevant or relevant to aggregation. But just because we think that doesn't mean it's true. And so we only got into LLPS indirectly, and still we're not proposing that synuclein itself is undergoing phase separation in this context. We're only really proposing that it's releasing vesicles from condensates that are formed from synapsin. And we find that to be very interesting. Also for tau, liquid-liquid phase separation, I'm much more interested in the idea that it's somehow functional, that it's related to promotion of tubulin polymerization. I think that's a very interesting idea that's out there, and it's not so clear to me where would tau droplets form on their own in cells, but it doesn't mean that it wouldn't happen. So Sunil Kumar, very nice work, thank you. In cells, do you think there's a possibility of oligomer formation that modulate the exocytosis process? I guess you could check this by staining with oligomer fibril-specific antibodies. All do you think synuclein is phosphorylated and add the effect of exocytosis? So a little bit along the lines of what Christian asked. Yeah, we've looked a little bit with antibodies that are specific to aggregates. So you do get a background staining by those. I think it's, whether they're staining oligomers or fibrils or what, I think is still an open question. So it's a good question. Part of our paper that I didn't talk about relates to synuclein co-localization with mitochondria, and we're following that up as well. There are interesting effects on mitochondrial function. And in that context, we think that serine 129 phosphorylation might be important. So I didn't have time to talk about that. So I think there is phosphorylation in these cells under condition of stress, but not very much under the conditions that we looked at here. So I'll say that. And then, did I get all the questions, or is there another one?
If you go back to the top, David, there are a couple that predate.
I also let in the panel, so we can ask Galina. Galina can ask the questions.
Yeah. Okay, very good. Galina, do you want to ask?
So I was wondering about the broken helix of the alpha-synuclein. Is it always has to be in between the vesicle and the membrane, or can it be seen on the singular vesicle, for example? Has it been shown? Yeah. And is there a different pattern of PTMs of these different conformational states? So I'm not talking about the fibrils, not during aggregation, but in actual conformational states, whether it's a straight alpha helix or a broken one, because you have this loop that can be accessed. Yeah. Thanks.
Sure, thanks. It's a good question. There are data there suggesting that the broken helix conformation can be formed on an isolated vesicle. And so Tobias Ulmer has such data. Some of the ESR groups have also suggested some data. So it's likely that that this state forms to some extent, that this linker is labile. And we also published a paper looking at the protein and in fluorinated alcohol, showing that this linker is sort of intrinsically weak and is not as solid as the rest of the protein. So I do think there's this state exists even when it's not bridging between two membranes to some extent. The only PTM we've looked at is tyrosine 39 phosphorylation, and that definitely has an effect. It's right at the linker. Most of the other PTMs are in the C-terminus, so those are less likely to have an effect there. Those probably affect protein-protein interactions, we think. Of course, N-terminal acetylation is critical to all this. I should have said that the protein in these our in vitro studies are now done pretty much only with N-terminally acetylated protein, because that's a physiologically relevant form, and that really enhances binding at the very N-terminus. We do think more and more that this N-terminal anchor is very nice work from Berthe's lab, Krageland, showing. There's other work as well showing that the N-terminus, the acetylated N-terminus, is sort of an independent binding motif, and the amphipathic helix is also a somewhat independent binding motif. We really think that these two motifs collaborate to give you the affinity that you need, and the helix 2 binding appears to be somewhat independent from the N-terminal binding. This is a section I didn't get to, and that makes sense also if you expect that region to bind separately to a different membrane. So I think that also works. But other PTMs that people have looked at, so there's lysine acetylation, there is glycation. Those are mostly thought to be associated with disease. The only functional one that I'm aware of in the membrane binding domain is Y39 phosphorylation. So.
Great, thank you. And especially with the relevance of the alpha-synuclein expression in the brain, because it gets expressed at a very specific time points. What we know from the finches, like birds, songbirds, that It starts being expressed during the song learning period. So maybe that's when you need that attenuation of the synaptic release to make sure that the memory actually gets consolidated. Yeah, very interesting. Thank you.
Thank you very much.
And David, there was an anonymous question near the very top of your list, which I can read so you don't have to look for it, which was just a very interesting talk. In the RBH cells, the staining looks similar to perinuclear agrosome formation, and there are 2 nuclei in the images shown, signifying that there is a cell division impairment upon formation of agrosome. In addition, there are nuclear puncti of the same staining, similar to other cell line models. And so then the question is, are these endosomes that somehow got relocalized to the nucleus, or are they nuclear or agrosomes that signify the prostatic impairment that leads to the endocytic impairment, and not necessarily a direct impact of overexpression of the alpha-synuclein?
This is a complicated question that probably requires a better cell biologist than I am. So the ERC is a perinuclear organelle, and so we don't think we're getting agrosomes there, even though it probably does look a little bit like agrosome formation. That's very typical of what ERC staining looks like. So it's a tubulo-reticular organelle that's near the nucleus, is how it's oftentimes described. We do occasionally see fluorescence in the nucleus, and so this, I think, supports the idea that synuclein was aptly named and that it does have a nuclear localization, and that's been seen by various groups over the years, and we just don't really know what that means. And so I can't really speak to what's going on, whether it's interacting with nucleolus, which is a liquid-liquid phase separated organelle, or whether it's interacting with nucleic acid, what some people have proposed. It's difficult for me to speak to that. I see a question in the chat from David Eisenberg. Thanks, David, for this fascinating picture of the function of alpha-synuclein. How do you envisage the mechanism of vesicle release from condensates by alpha-synuclein? Yeah. So at this point, we think it's likely that synuclein is competing with synapsin for binding sites on the vesicle surface. And so when synapsin binds to those sites, since synapsin is via C-terminal disorder domain clustered in condensates, then the vesicles are trapped. And we think synuclein basically displaces synapsin from the vesicle surface and releases it, but we don't have data to support that. It's just that the model that we have right now. So it's an interesting question and will be interesting to see. And it's relatively easy to test in principle if Topo weren't leaving the lab, unfortunately, which happens. So.
And then we have a comment from an anonymous attendee, which was, hi, David, great talk. There's a paper by Frost Group at UC San Francisco on LEM2 and CHMP7. LEM2 has an intrinsically disordered region, and they show it phase separates. LEM2 also has phosphorylation sites and phase separates on microtubules. Could be worth looking at.
I would agree.
Let's see, and then it looks like we have one more question. It's from Sarat. Hi, David. Beautiful talk. Do you have any evidence of the role of the C-terminal part of alpha-synuclein? What's the role of this acidic segment?
Yeah, that's a tough question. We're looking a little bit more carefully now into interactions with snare proteins. So there, probably many of you, at least in the synuclein field, remember that Bourret and Sudov published a paper suggesting that synuclein interacts with VAMP2, which is the vesicular snare protein. We looked at that early on by truncating the C-terminus, which is where the interaction site was supposed to be. We did not see any effects on function in our functional assays. We didn't see any interaction in vitro that was obvious. So we kind of said, nah. There was a recent paper by Ed Chapman in collaboration with the guy at UCSD, I can't remember his name right now, I apologize, suggesting that site might actually be not at the very end of the C-terminus, but actually might be sort of more around residues 90 to 100. And so we're having another look at that to see whether that's something we missed. But we generally think of the C-terminus as a protein-protein interaction motif. Certainly with RABs, that was shown by Marcus and Christian, and anyway, it was shown in a nice paper, and we're interested in that as well. It's been proposed, and the amazing thing about synuclein is that many proteomic studies, proximity studies, et cetera, there are actually very few authentic binding partners for alpha-synuclein that have been characterized, whereas there must be more out there that we just don't understand. And that would be also extremely useful, I think, to having a better idea of what it is that it does. So good question, but I don't have any profound answer.
So David, if I could ask a question, you mentioned this triple knockout mouse that had been studied by another. What actually happens to those mice? Clearly they're not embryonic lethal, but do they live a normal lifespan? Do they have this defects in? Yeah,
that's a very good question. So there have been a number of reports on knockout mice. They don't all agree with each other, but the triple knockout mice, it would be nice if they had Parkinson's disease for people who are interested in function like me, but they don't really have anything that's obviously Parkinson's disease, but they do develop neurological dysfunction at old age. So around, I think, 18 months or 2 years of age, they have neurological deficits. So that suggests that alpha-synuclein does do something, which of course is nice to know, but it doesn't provide a lot of clarity in terms of linking it to Parkinson's disease. So you know, the idea of whether function is important in amyloid disease in general, I think is, you know, whether, you know, in Huntington's or in tau or in alpha-synuclein, you know, most people would say no, but there's a, you know, there's a sub-community of people who say maybe at least, if not yes. And I think there's still some room for that.
Great. I think we've hit all of the questions. And so why don't we, let's see, there's something in the chat here. Why don't we open up the floor? So if there are any questions from the attendees for Lucia that you didn't get the chance to ask, or any more from David.
I had a really naive and ignorant question for Lucia if nobody else jumps in.
Go ahead.
You know, so you mentioned in many of your spectra that you were determining whether the cysteines were oxidized or reduced, but I thought, and you know, maybe I'm wrong, but I thought that you really needed the carbon chemical shifts for that, the alpha carbon or beta carbon chemical shift for that, and it looked like you only had N15 labeling. So were you just doing that based on comparison with reduced and oxidized samples in vitro, or how could you actually tell whether your cysteines were reduced or oxidized?
No, yeah, that is a good question, and actually I should say that is I'm surprised that this is the first time I've been asked this question. Indeed, we normally use the carbon-13 chemical shift of cysteines to say whether it's oxidized or reduced the cysteine. But in this specific case, SOD is the states, the spectra are very similar in solution and in vitro and in the cell. And so then by the comparison, while even the nitrogen 15 chemical shift, they changes a lot. So then we can clearly monitor the status of the cysteine. And obviously they are in slow exchange. So then if we have a ratio in other proteins, when we have some fraction oxidized and fraction reduced, we can clearly state, but by themselves it's true, the nitrogen 15 chemical shift of the cysteine are not a reporter of the oxidation state of the cysteine. But having the assignment, so I come back to the initial comment or some question and ask, having the assignment of the different states, we can clearly say that, so it's an indirect, but it's very solid because there are very clear differences. Also, I want to say that in general, for example, enzymes, they are soluble proteins. They are not expected to interact with other components. So the cellular environment affect the function of the system, but in terms of structure is maintained, is not different. In other cases, for other proteins, then we have differences. So in this case, the spectra are very, very close and is easy to, but you are absolutely right. And keep in mind, I have been shown this data many times, and you are the first person to ask me this question.
Thank you.
Yeah.
Lucia, there's a question from Luis Del Pozo. I know you answered the question by typing it. Could you read out the question, or could you or Claudio, and then answer that so that it can be recorded in the...
No open questions. I don't see any questions.
In the Q&A, if you go to the answered column, you will see that.
Okay.
Oh, okay. Oh, okay. Yes, I answered by typing. Yes.
Yeah. Could you, one of you, read out and then record the answer?
Where is because sorry,
the fourth one from the top.
Yeah, 1/3 of the list.
Okay, that was the question was regarding some specific cells and the mesangial cells internalized in the amyloidogenic light chain. And so actually what it was asked is, could this type of cell a good model for in-cell NMR analysis for characterizing misfolding and amyloid aggregation of light chains? Now, beside that, the protein I've been studying, there is the light chains, I mean, just individual proteins. To study overall aggregation over time, there is an issue because, as I said, for the time being, we use characterize and all the other people just the cells inside the NMR tube, and then cells would will be alive for 2 hours or so. And this is a too short time to have aggregation. In the specific case of SOD, we detected aggregation over months. So that is a problem of the time. If you want to monitor the setup of the aggregates and the formation of the aggregates, it might be that the bioreactor, still we were talking about hours or something, like we tested up to 50 hours. Still we can see some initial step of aggregation. This is regarding the time frame for the aggregation. Regarding the aspect of using other cells, and this is what I answer to this question, is that for what we try to have a reasonable expression level of proteins in different types of cells, and we have been successful regarding human cells with ELA cells. In other cases, we have expression, but at solo level that are not yet detectable with NMR. But even in my case, even more, we are not a cellular biologist. So for the time being, we have been able to do this on these cells. So X cells, CHO, ELA cells, and so then is a problem of reasonable level of proteins to be detected by NMR. If we inject, that is another story. Then we can use any other type of cells. So what for the time being, we were concentrated working on proteins directly produced in the cell. Eliezer, Claudia.
Yeah, I have a question to both David and you, Lucia. Do you think you might conceive a meaningful experiment by using in vivo in cell NMR to look at the binding of synuclein to cell membranes, to vesicles and stuff? Or that is actually forbidding anything because once synuclein is bound, you don't see it?
Well, go ahead, Lucia, first.
No. I mean, you know, as I said, we can detect the signals of synuclein in the cell. So then in the X cells, so it might be that there is a fraction that is bound, but overall we detect the signals. So I don't know what, I mean, this interaction with the membranes that David has shown, it should be fast on the in terms of timing. It doesn't require, it should be as protein is produced, it should be bound there. So we didn't see particular effect. So probably might be the types of cells, because when he was talking, David was talking on specific type of cells, and indeed, I mean, has been observed, I think, by Phil, that only when it's inserted in synaptic cells, so that you see the aggregation. In all other cells, so that it behaves normally. So it might be that are very specific, not just in general plasma membrane. But I don't know, what do you think, Dave?
Well, you know, there's a history of people trying this already. And so Phil Solenko, as you mentioned, looked at this, and you know, they saw largely soluble protein with some indication of transient interactions with hydrophobic species, and they didn't, you know, they could model this using BSA. And then there was a paper from Sebastian Hiller more recently where they claim that there are interactions, chaperone interactions with the N-terminus, and the N-terminus is, you know, perhaps the most important membrane binding motif. And so that could be a potential explanation why you don't see the protein associated with membranes, because the chaperones are holding on to the N-terminus, and so that part actually usually you do see intensity decreases for the N-terminal 10 residues or so, oftentimes in cell, and not for the rest of it. Also, as Lucia said, you really need to have synaptic vesicles, those are synaptic vesicles. So hex cells are not really secretory cells, I don't think. I don't know a lot about their membrane trafficking. So that's also interesting. So there's a lot, I think, to be done still in cell NMR with synuclein, and it's not something that we do a lot of. We did a little tiny bit in bacteria way back when the N-terminal acetylation and the tetramer story was relevant. But I think there's still a lot to explore. And I know Phil has worked very actively in this area, and as I said, Roland and Sebastian also contributed. Although the chaperone story is a bit murky because it's not clear. They're bacterial chaperones, mammalian chaperones, so it's there's more to be done there, I think.
David, we just got another question, which is it's very interesting that you mentioned a few times the similarity between alpha-synuclein and tau in terms of IDP structure, tendency to form aggregates, and liquid-liquid phase separation and normal function in disease state. Can you elaborate that similarity a little bit more and their differences with FUS and TDP43?
Well, this is a big question, but I can do a little bit. I think it's an interesting question. So FUS and TDP43 really fall into the category of proteins that have really low complexity domains, prion-like domains, and also RNA binding domains. And so this is a somewhat different category, I think. And alpha-synuclein and tau, especially alpha-synuclein, if you look at it on the so-called Uversky plot, it's really barely even an IDP on that kind of plot. It has a fair amount of hydrophobic character, which allows it to bind to membranes, and it's not a low, it doesn't have a lot of aromatics, but it's not really a low complexity sequence. So they're kind of different. And tau, I think, probably looks more like synuclein. I haven't looked at it on that kind of plot. It does have tau is big, so it has different regions, but there's a proline-rich region, but there's quite a bit of amino acid diversity in tau as well. So I think, the low complexity domains, they probably, they're not my area of expertise, but they probably don't interact with much beside themselves, I think, unless you have things like prothymosin alpha, where you have sort of this amazing electrostatic interaction with histone tails. Whereas alpha-synuclein and tau clearly do, tau binds to microtubules and synuclein binds to vesicles. And so there's sort of two different classes of IDPs and of amyloid forming proteins. And then, of course, you have proteins that are perfectly well folded to form amyloid as well. So I do think that both for tau and for synuclein, membrane interactions are particularly interesting. So, it's an area that obviously we spend a lot of time thinking about. We showed early on that tau also binds to membranes. We think there's a similar type of process where you get amphipathic helix formation right next to the amyloidogenic motifs in tau, the PHF6 and PHF6 star motifs. So we think you may have a very similar process. In fact, it would be if life is already finished with alpha-synuclein, Christian, maybe you want to have them look at this with tau, or we don't have the bandwidth for it. So somebody could probably make an interesting story out of that potentially. So there are some interesting similarities in terms of liquid-liquid phase separation. Again, I think my feeling is that you can induce almost any protein to. It's Maggi, I think, who had. I don't know if that paper is out yet or not, but I think his group went through like 50 different proteins of all sorts and showed that they could find conditions to get condensates for all of those proteins, basically. So I'm not so sure if there's something special there for synuclein and tau. Thank you for the question, though.
Great talks from Lucia and David. I liked it very much. So I would like to continue on that in-cell NMR work from Selenko and Danilo EPR and Selenko using NMR spectroscopy. Do you think that they reported basically the monomer, disordered monomer is the state for synuclein inside the cell? So it's likely that they're not seeing the bound ones like what you guys are seeing, membrane bound or bound to other hydrophobic compounds, so which will make it bigger species which not tumble fast enough for detection. So can you comment on that?
Well, I think it's complicated, I think, in the sense that I think Phil's experiments were very profound, but at the end of the day, this was still protein that was reintroduced into the cell. So it's a little bit difficult to know, but yeah, but I don't know. I mean, we've always been puzzled by the fact that the protein looks like a soluble monomer rather than, there's a lot of membrane in cells. You would have thought, even though the binding to the plasma membrane is much weaker because there's a curvature dependence to the binding, so it doesn't bind well to other membranes. I would have thought that there would be a lot of protein that's actually bound. So maybe it's because these chaperones interfere with that binding. I think there's some unpublished data. I think in yeast, maybe something else goes on, and maybe the different chaperones. I don't know. Lucia, what do you think?
Yeah, that is, as you said, that this is a protein that has been produced in coli, and then has been inserted in the cell, and then in the insertion treatment, you might alter or affect the properties, one aspect. Second, I mean, this is one point. My experience, we characterize alpha-synuclein in cell, a different magnetic field, including the 1.2 gigahertz. And we see that the soluble protein, but as I said, is a specific type of cell, and that is not, I mean, alpha-synuclein has most likely a very specific different behavior depending on the cells. And I think we have to find a way to have reasonable amount directly expressed in the cells that are relevant, the synaptic cells, so that the neural cells. So then I think that would be the way, because if you insert, you might have different methods, can be milder or not, but you have some effect, and on the folding, you have an effect. Some cases you try to insert proteins and you unfold the protein if you use that approach. So
sorry, go ahead.
No, this is why I think it might be a correct behavior. I personally don't know. I haven't analyzed the alpha-synuclein. I've been not working on alpha-synuclein. I have only used as unstructured protein to monitor the different behavior between the unstructured and structured protein. So then for me, I keep as unstructured. And in all my experience has been behave as a monomer unstructured protein. And not binding on, not being stick on over membrane, because the intensity is more or less what I expect for the amount of protein that induce the production, and that we monitor through Western blot how much is in there. So I can talk about alpha-synuclein in terms of physiological behavior. I don't know.
It is worth mentioning that in some sense the NMR measurements are consistent also with the fluorescence data. So if you look, even in our cells, when we fluorescently label the protein and look at the cells, most of the protein is in the cytosol. And remember, if the data I showed you in the neurons, most of the cell protein is not on the membrane. So the interaction with membranes is pretty weak. And in our model, the only tight binding mode is when you bridge between membranes, and that's going to be a relatively rare scenario. So if you look at a synaptic terminal, there's only going to be 10 vesicles docked at any one time. So maybe you could have 50 molecules of alpha-synuclein, let's say 5 per vesicle, 10 per vesicle, 100, but most of it doesn't have a high affinity binding site. So this interaction with the vesicles is much weaker. And you don't see synuclein parked on vesicles in cells. You don't see synuclein parked on membranes by fluorescence either. So I don't think there's a big discrepancy between the NMR and what it actually does.
And Bikash has a question. So continuing on that, so I agree on the binding affinity, but it could also be depending on the membrane composition. For example, anionic lipids have been shown to promote the binding affinity for synuclein. So I don't know how maybe cholesterol can inhibit as well. So I do not know. So how much is that would play a role? Okay, Bikash, yeah, go ahead. So
I was just going to say cholesterol is an interesting story, and Juliana has a nice paper on that recently, looking at effects of cholesterol, and it actually affects binding of part of the membrane binding domain in a way that again supports the idea that this thing can reach out to other membranes. It's something that we wanted to do also. And cholesterol is tricky to work with, so there's actually a lot of technical issues getting cholesterol into liposomes, but it's also a nice paper. So sorry, go ahead.
Bikash, you have a question?
Yeah, I have a question for Lucia. So it's a very nice talk. I have a very fundamental question on the in-cell NMR. Like, I think like when you do like overexpression of the protein to do in-cell NMR, so eventually we are increasing the local concentration of the protein inside the cell. So that is, I think, one of the major limitations that we all know. And another thing is that like when I think like you are looking at the SOD1, like we know that the SOD1 has been reported to have localized in mitochondria, nucleus, as well as in cytoplasm, right? And we already have discussed extensively that like depending on like what are the binding partners, some are visible and some are not invisible, right? And all of your data that you are showing is actually the average, because in NMR we are getting the average spectrum. So we don't know actually in the overexpressed cell how much of the protein is actually in the nucleus, how much are in the mitochondria, and how much are in the cytoplasm. So how do you actually comment these limitations?
Yeah, I mean regarding the cell distribution, cellular distribution, this is something that we for a number of proteins we characterize, because we separate the different cellular components and we measure, we estimate, let's say, the concentration of the protein, the amount of protein in the different cellular compartments. We have done also in organello NMR. So for some proteins, we have characterized and collected the spectra on just an ensemble of mitochondria. So we have extracted the intact mitochondria, put them in the NMR tube, and collected the spectra of mitochondria. Overall, the volume is much less of the mitochondria with respect to the entire cell that is cytoplasm. So depending on the type of proteins, the proteins is either only in the cytoplasm or is present in other cellular compartment. In the nucleus, there is not much. The nucleus has continuous exchange with the cytoplasm, because we have tried to characterize some protein or to have extract the nuclei and put the nucleus in the NMR tube, but then there is the nuclear pore is quite open and the proteins get in and out. While when they are in mitochondria, they are stuck there. So just to conclude, we fraction the cells and we estimate the amount of protein in the different compartments. And then also we can characterize directly the proteins in mitochondria. We don't detect the signal of the protein in mitochondria because they are very low amount. And in the case of SOD, the spectra are more or less the same of the one of cytoplasmatic. The structure of SOD is always that.
Okay. I'm noticing there's a couple of comments in the chat that I should address from Galina. Hi Galina. I didn't put together you and you, but we know each other well indirectly. So Buchmann, yeah, Vladimir, of course, has a triple knockout mice. People are asking about the triple knockout mice, and Sudaf also has a triple knockout mouse, and they don't always see the same thing. So it's actually interesting, but there are background effects there that remain to be understood. And then Galina, you ask whether there are known truncated alpha-synuclein species, and I'm sure you probably know better than I do, but there are in fact isoforms of alpha. First of all, there's truncation, enzymatic truncation of alpha-synuclein in disease, absolutely. So C-terminal truncation, there have been various C-terminal truncations shown by mass spec, and the more you lop off, the C-terminus is very negatively charged, so it's sort of an intracellular solubilization motif in a way, and the more you lop it off, the more the protein is aggregation prone, so that C-terminal truncation probably is important. But there are also isoforms, not truncations, enzymatic truncations, but isoforms of alpha-synuclein that are missing parts of the C-terminus, and that's sort of not really widely appreciated. And I don't think we have a good feeling for how much they are expressed, when they're expressed, how much of them are around, and what their role is either in function or in disease. So that's actually quite interesting as well.
Looks
Yeah.
like there are no other
Yeah.
questions. Thank you all very much. Thanks, David. Thanks, Lucia.
Thank you.
Thanks, Dan, and thanks, Claudio. It's really lovely.
Thank you.
ZOOMinar.
Thank
We
you.
all loved
Thanks
it.
a lot.
Yeah,
Very
thank you.
good.
Now we can
Thank
go
you,
and have
everybody.
wine if you want.
Nice to see
Bye.
everybody.
Thank you for coming.
Yeah.
Yeah, really great talks.